Good morning, students. Today we will do the topic photorespiration. Now we have seen in our previous classes, studied in detail about photosynthesis, that the first step of dark reaction in photosynthesis is combining of carbon dioxide with ribulose biphosphate, which results in the formation of phosphoglyceric acid. Now this reaction is inhibited if there is a high concentration of oxygen. So a high concentration of oxygen inhibits photosynthesis. The reason is that the enzyme RuBP carboxylase, ribulose biphosphate carboxylase, also combines with oxygen, helps in the combination of oxygen with RuBP. It has the ability to catalyze both the reactions. So if concentration of oxygen is high, then it will not help in fixation of carbon dioxide. Instead, the enzyme is called ribulose biphosphate oxygenase, and it combines with oxygen to form phosphoglycolate and phosphoglyceric acid. I think I will write it and explain it to you. We are doing the topic photorespiration today. Now you have seen that in photosynthesis, I was comparing the two. Carbon dioxide combines with RuBP in presence of rubisco, that is ribulose biphosphate carboxylase enzyme, and forms phosphoglyceric acid. Now if oxygen concentration is high, then photosynthesis is inhibited. Photosynthesis is inhibited because the same enzyme combines with oxygen, helps in the combination of ribulose biphosphate with oxygen and forms phosphoglycolate and phosphoglyceric acid. It has more affinity for oxygen, and in that case it is called RuBP oxygenase. Instead of carboxylase, the enzyme is called oxygen. Here it is helping in the combination of carbon dioxide with RuBP. So the enzyme is called RuBP carboxylase. This is formed here, phosphoglycolate. Now this is called photorespiration, and photorespiration occurs in three organelles. These three organelles are chloroplast, peroxisomes, and mitochondria. Now this phosphoglycolate then changes into glycolate. Glycolate then changes into glyoxylate, and glyoxylate then changes into glycine. So reaction till here takes place in the chloroplast. This reaction takes place in the peroxisomes. Then glycine enters the mitochondria. Glycine enters mitochondria, it forms serine, and carbon dioxide is removed. This reaction takes place in the mitochondria. I am just telling you in brief the sequence of reactions that occur in photorespiration. So the first step is changed in photosynthesis. Why photosynthesis is inhibited in presence of high oxygen concentration? Because the activity of the enzyme is changing over here. Instead of carboxylase, it starts functioning as an oxygenase and helps in the oxygenation of ribulose biphosphate, and the product is phosphoglycolate, which is then converted into glycolate, then converted into glyoxylate, glycine, and glycine changes into serine and carbon dioxide. So this is called photorespiration, and this was discovered by the scientist J.P. Decker. The scientist J.P. Decker discovered this process and he called it photorespiration because it is occurring in the presence of light. This reaction is a light dependent reaction. So this is called photorespiration. I will just read it out to you. A high concentration of oxygen inhibits photosynthesis. This is due to the reason that carbon dioxide fixing enzyme RuBP carboxylate not only accepts carbon dioxide but it can also combine with oxygen. Since the reaction is an oxygenation reaction, the same enzyme is called RuBP oxygenase. The sequence of events is that instead of carbon dioxide and RuBP, instead of two molecules of carbon dioxide and RuBP forming two molecules of phosphoglyceric acid, oxygen combines with RuBP, forms phosphoglycolate and phosphoglyceric acid. This phosphoglycolate then changes into glycolate, then changes into glyoxylate. This reaction, change of glycolate into glyoxylate, occurs in the peroxisomes, which then further changes into glycine. Glycine changes into serine and carbon dioxide. The process is known as photorespiration. It was given by the scientist J.P. Decker. Photorespiration is a light-dependent uptake of oxygen and output of carbon dioxide, because carbon dioxide is a product. This is being produced. So this is output of carbon dioxide and uptake of oxygen. The carbon lost, this carbon that is lost, cannot be retrieved. So there is loss. Here carbon dioxide is being fixed in the form of food. Here carbon is being lost. The carbon lost is not retrieved. The plant may lose about 25% of the carbon dioxide, which is fixed during photosynthesis by this process. No energy-rich compound is produced. Rather, there is a wasteful loss of ATP and NADPH2. When I write the full sequence of the reactions, you will see ATP is utilized in the reaction and NADPH2 is also utilized. So there is a wasteful loss of ATP and NADPH2. It involves three organelles, the chloroplasts, peroxisomes and mitochondria. An increase of temperature leads to more photorespiration, and this means there is more loss of photosynthetically fixed carbon. It occurs in light and it has no relation with the normal respiration. It decreases the yield of the C3 plants which grow in tropics. But it has a useful role also. Photorespiration is useful in protecting the plants from photooxidative damage under conditions of high light and limited carbon dioxide supply, so that the plants do not get damage in high light and less carbon dioxide concentration. The excess light energy can cause damage to the plants. Photorespiration, which means oxygenation of ribulose biphosphate, utilizes part of the light energy and saves the plant from the photooxidative damage. Photorespiration is also called as photosynthetic carbon oxidation cycle. Photorespiration is called photosynthesis carbon oxidation cycle. So this is what I have told you about in brief about photorespiration. Now I will draw the cycle, an outline of the cycle properly, showing you the three organelles and the sequence of reactions that take place during photorespiration. After that, we will do the factors that affect photosynthesis. Now, the first reaction is oxygen combining with ribulose biphosphate. It forms phosphoglycolate and PGA. This phosphoglycolate then changes into glycolate. This reaction occurs in the chloroplast, first organelle. Then glycolate moves into the peroxisome. Suppose this is the peroxisome. Glycolate moves into the peroxisome, changes into glyoxylate. This glyoxylate then changes into glycine. This glycine is then transported into, this is the third organelle, mitochondria. You know, mitochondria has cristae like this. So just to show the difference between chloroplast and mitochondria, let us say this is mitochondria. Glycine in mitochondria changes to serine and carbon dioxide. This serine then changes to glycerate. Here NADPH2 is utilized. Wasteful loss of NADPH2 and ATP is also used. So we said there is wasteful loss of ATP and NADPH2. And here we see oxygen. This is mitochondria. Why we are calling it photorespiration? Because respiration, the site of respiration is mitochondria. This is occurring, it is using oxygen and carbon dioxide is produced. So this occurs in the mitochondria. We can write here oxygen is used up and carbon dioxide is produced. So this is photorespiration, which occurs in light. It is a light dependent reaction. And we have studied the useful role of photorespiration, that it helps in protecting the plant from photooxidative damage. Photooxidative damage is reduced in the plant. This is the useful role of photo. So this was about photorespiration. Now the next topic that we are going to do, and with that we will complete the chapter on photosynthesis. The last topic is factors that affect photosynthesis. So I am going to erase this and we will begin with factors that affect photosynthesis. Now, the rate of photosynthesis is influenced by a number of environmental factors like light, availability of carbon dioxide and water, temperature, and a number of genetic factors which are called internal factors. These are the age of the leaf angle, leaf orientation, etc. Now, photosynthesis is not affected by all environmental factors at a given time, at the same time. When several factors affect photosynthesis, any biochemical process like photosynthesis, Blackman's law of limiting factors comes into effect. The law proposed by Blackman on limiting factors, that is taken into consideration. So the rate of photosynthesis is governed by the law of limiting factors, and according to this law, when a chemical process is affected by more than one factor, the rate of process is limited by the slowest step in the pathway, or that is called the most limiting factor. One that is slowest or the lowest out of a number of factors, the one that is the lowest or the slowest, that is going to affect the rate. And in this case we are talking of rate of photosynthesis will be affected by the one that is the lowest. This is the law of limiting factor proposed by Blackman. It means that at a given time, only the factor that is most limited will directly affect the process. So we are talking about Blackman's law of limiting factor. They said number of factors like carbon dioxide concentration, availability of light, temperature, etc. Availability of carbon dioxide, light and temperature. These are the main factors that affect the rate of photosynthesis. So it will be affected. Rate of photosynthesis is affected by light, carbon dioxide concentration, temperature, etc. There are other genetic factors, but these are the main, and even water. So these are the main factors. So this rate is actually affected by the factor that is lowest or slowest. This is called law of limiting factor. We can say it means Blackman's law says that at a given time, only the factor that is most limiting will directly affect the process. And we can study Blackman's law of limiting factor by performing 3 simple experiments. How one factor can become limiting. This we show light intensity on the x-axis and rate of photosynthesis on the y-axis. We'll see with increasing light intensity, rate of photosynthesis increases in experiment 1, or let us say in experiment A. This increases, and when this is taking place at atmospheric carbon dioxide concentration, that is 0.04%, and at temperature 15 degrees Celsius. Now if concentration of carbon dioxide is increased and temperature remains the same, then also rate of photosynthesis increases if carbon dioxide concentration becomes high. The temperature is 15 degrees. This was seen in the second experiment. Now in the third experiment, if now this is constant, here we are taking into consideration carbon dioxide concentration. Now with increase of carbon dioxide and temperature, we will see more carbon dioxide concentration and even temperature is increased, then only the rate of photosynthesis will increase. temperature has been increased. So in experiment A, we see the light intensity is increased, the rate of photosynthesis, we are increasing the light intensity, the rate of photosynthesis is increased till both temperature and carbon dioxide become the limiting factors. In experiment B, increase in carbon dioxide concentration. We have increased the carbon dioxide concentration from experiment A to experiment B. And this increases the rate of photosynthesis till temperature becomes a limiting factor. Now temperature is also increased and carbon dioxide is also increased. Now rate of photosynthesis increases. If both are increased, now light becomes a limiting factor. In this case, carbon dioxide was a limiting factor. In this, temperature was a limiting factor. In the third experiment, light is a limiting factor. So this proves Blackman's law of limiting factor. Then we come to the factors that affect photosynthesis, the external factors. Light. The quality, intensity and duration of light which is received by a plant greatly influence the rate of photosynthesis. Light quality. Photosynthesis takes place in the visible part of the electromagnetic radiation, and we have already studied that blue and red regions of the visible spectrum are the most effective, whereas the green is the least effective. Then coming to intensity of light, there is no photosynthesis in the absence of light. As the light intensity increases, photosynthesis increases proportionately until the saturation point reaches. Saturation point is that point beyond which an increase in light intensity does not cause any further increase in the rate of photosynthesis. After saturation point, some factors like carbon dioxide may become limiting. Very high light intensity may reduce the photosynthesis as it may lead to bleaching or destruction of chlorophyll. This phenomena is called photooxidative damage. Now, carotenoids play a protective role by absorbing excess light and diverting it away from the chlorophyll. They also help to detoxify the bad effect of activated oxygen on the chlorophyll molecule. Therefore, we say carotenoids act as antioxidants. Then, duration of light. Plants getting an average of 10 to 12 hours of light per day, they show higher rate of photosynthesis. Next is temperature. Temperature has less effect on light reaction, but dark reaction, which is enzyme catalyzed, which is controlled by enzymes, is greatly influenced by temperature, because we know the enzymes are proteins and proteins get denatured at very high temperature. So even low temperature, the activity of the enzyme will be reduced, and at very high temperature, the activity of the enzyme will be affected. The temperate C3 plants work best at optimum temperature of 25 degrees Celsius, while the C4 plants of the tropical regions, they need a higher temperature and the optimum temperature is higher for them. And a rise above 35 degrees Celsius leads to decreased rate of photosynthesis. Then the third factor is concentration of carbon dioxide. Carbon dioxide is required for dark reaction. The land plants absorb carbon dioxide through stomata. So when the stomata close, the rate of photosynthesis decreases due to non-availability of carbon dioxide. So the plants that are submerged in water, they absorb carbon dioxide in the form of carbonates or bicarbonates, which are dissolved in water through epidermis or the mesophyll cells. So we have seen the effect of light, how quality of light, intensity of light, duration of light has an effect on photosynthesis. Then temperature, how it influences photosynthesis, and lastly the effect of carbon dioxide concentration on photosynthesis. With this, we complete the chapter on photosynthesis today, and your half-yearly syllabus includes all the chapters that we have done till now. Is that clear? Now I will be giving you a few assignments based on this. After that, we will do some small topics, though they will not be included in the half-yearly exam, but part of it which you have done may be asked in the exam. As and when we do that topic, it will become clear to you. So that's all. Good luck to all of you. Study well.